I appreciate that, and if we could ask the next panel to come up, we'd like to hop right into it and get back on time, and so sure all track all these folks down at lunch. So Mike, if you can gather your panelists, I think we're going to be good to go. Let me make sure we're in the right scenario. Nope, that was Jay. Break? Nope, no break. You just had your break. Just then. It's over. Okay. So we're moving to the next panel. I guess, again, thank our last panelists, because it's really, to me, very reassuring that sort of all of these ongoing community-based efforts and local efforts really reflect, really I can trace it back to some of the earlier discussions we had about the oceanographic work that's being done by WHOI. And so really kind of connecting all of these efforts from kind of the local place-based scale up into the research and other objectives is really, I think, an important aspect of this. So with that, I will turn this over to Mike Crowley. Thank you.
Good morning, everybody. Good to see everybody here today. I'm Mike Crowley. I am from Rutgers University, Center for Ocean Observing Leadership. A lot of the work we do is in ocean observing, physical biology and chemistry side of things. My job there is technical director, which means I deal a lot with the operations of what happens over there, of gathering data and making data available online as fast as we possibly can. A big chunk of my job is working for MARACOOS, Mid-Atlantic Regional Association Coastal Ocean Observing System, right? Rolls off the tongue. It's the real-time observation kind of counterpart to MARCO, right? The policy piece for the Mid-Atlantic. This is looking at real-time ocean data. We have a Oceans Map is a portal that we use to look at all sorts of real-time data that we collect and that the government collects and states collect and everybody. It's all put into one spot. It's kind of the counterpart to the MARCO policy and planning portal. It's on the real-time side of things. So I'm probably the perfect person to host this session, as I'm not a biologist. My background is heavy on the physical oceanography side of things, so Gulf Stream ring and meander dynamics, coastal upwelling, also all the physical drivers of what happens in the ocean, but of course the physical drivers help drive what happens in the biological and chemistry sides. So I've worked heavily with the biologist over my, 34 years in oceanography. So we're going to get started. Last session before lunch. I know people are probably hungry. So we have three awesome young women here today, and I'm going to start off the first person here today, Grace Saba. So Grace grew up in the mecca of marine biology in Henryville, Kansas, and went to UC Santa Barbara, did her undergrad there, and then went to VIMS to do her PhD work, and then she joined Rutgers, and I've known Grace for 14 years. She joined Rutgers back in 2010, and her background is marine organism ecology and physiology, and looking at the physical drivers, right? The physical drivers of what drives the biology on the coastal zone. So, Grace, take it away.
Way down. Am I? Oh, perfect. Here we go. All right. Hi, everyone. It's nice to see familiar faces. I'm going to try to get through my presentation in the allotted time because I know we are the one session blocking you from lunch. So I just want to first acknowledge a collaborator that I've been working with a lot, Josh Kohut at Rutgers, and he's a physical oceanographer. I'm a biologist, so it's a good collaboration because we're using these gliders in a lot of different projects now to help kind of understand where and why organisms are where they're at in the ocean. So we use these eco-gliders as a tool, and I'll start by saying why, and why we want to use these in the Mid-Atlantic specifically, and that's because it's such a complex region. It's highly variable in time and space because there's just a lot going on. We have water from Labrador Sea coming down. This year we have a lot of that water coming down. It's so cold out in the oceans, particularly in the bottom waters right now. We have the Gulf Stream coming up north, which is completely opposite of the Labrador water. So it's warm, it's salty, and we're right where those two water masses mix. We have freshwater inputs, we have storms, so it's just highly complex and variable. So we need to make sure we're observing that variability at the right resolution. We're also situated in one of the most rapidly warming regions of the ocean. We sit right in the U.S. Northeast Shelf. These are ocean trends from 1993 to 2020. We're warming quite rapidly, and that's really, we're thinking that's because of the more northward motion of the Gulf Stream, which is very warm, and it's warming our coastline. And then because we have all these, we have a lot of variability, we have a lot of long-term change, the organisms respond very rapidly to that variability and that change. So, we have, the biology and the physics are very tightly coupled in our system and globally. And this is just showing some trends of fishes through time, looking at how fishes their distributions are moving poleward and into deeper water. Because of this kind of ongoing warming of our oceans. So we have that going on top of just shorter term variability in time. And now we're also changing the way we're using our ocean. There's a lot of plans for development of offshore wind in our region, and the reason for that is to mitigate that long-term climate change. But it's a lot of development that's going to happen in our region, and we just want to make sure that we have a lot of baseline information to understand that natural variability before we have these platforms in our ocean to better understand how they might impact organisms and other things, other oceanographic processes over time. So there's a need to establish a baseline that we can then assess that change in climate, but also change from things like development of offshore wind. So we use gliders. Let's see if this video will work. Oh wow, it's not working. Can I? Oh well. Anyway, that's unfortunate because it's a really cool video, but it shows how the glider works. But they're 1 meter long vehicles. They're autonomous underwater vehicles, so we can control them from the lab at Rutgers. We have several gliders in our program, but the benefit of these gliders is that they can sample from the surface to the bottom, so we're getting data at really high resolutions throughout the water column. They have wings which propel them forward, so we're getting, we can go about 20 kilometers per day on these gliders and fly them along tracks. They're sampling every 2 seconds, so it's super high resolution. And, so we're getting resolution both in time and space. They're relatively cheaper than ships. Ships can cost thousands to $10,000s per day and use fuel. So these are cost effective, and we can use them for a lot of different applications shown here. But again, the high resolution sampling, we can keep them out in our coastal ocean, we usually keep them out about a month at a time. Then we can recharge the batteries and put them back out. So that has a really high temporal coverage and gives, again, that spatial coverage. We can put them out in storms. We have a whole storm program at Rutgers, putting gliders out in hurricanes and in the path of hurricanes to better understand how the ocean impacts hurricane intensity forecasts, for instance. They're good for augmenting vessel-based surveys, and we're hoping that they'll be able to navigate wind farms because they are small and we have at least some control over where they're going. But the really fun thing about these gliders, because I'm a biologist, is that we can have a lot of different sensors integrated into them now. There's been a lot of advancement, particularly in the biological sensors, so that's really why I'm excited. But again, they're measuring the physics, so we get temperature, salinity, we get chemistry, dissolved oxygen. We have pH sensors on gliders now, so for ocean acidification applications. And we can measure biology, so we can look at biology all the way from phytoplankton using optical sensors to pelagic fishes and zooplankton using active acoustics. And then we have passive acoustic sensors, so they're listening for things like marine mammals and fish detection. So any fish or elasmobranchs that are tagged, we can hear them, or turtles, we can hear them with our gliders if they pass by close enough. So again, all of these data are combined when you can put these gliders out, and so you're getting the physics, the chemistry, and the biology all at the same time. So I just want to highlight 3 eco-glider programs that just Rutgers has been involved with in the past few years. And the first one has been a really collaborative partnership between several academic universities, but also the Ocean Acidification Program and IUS, which is the umbrella of MARACOOS, which Mike talked about. But really, it was meant to put gliders out to investigate spatiotemporal dynamics and drivers of carbonate chemistry in our region. So where are we seeing potential spots where ocean acidification is bad, right? Or where do we see repeated hot spots of ocean acidification? And that has implications for management, right? What organisms are in those locations, and do we have to think about how they're gonna respond? And then we have an ongoing project now with New Jersey RMI program, which Katrina, that was such a nice introduction she gave me this morning. I was like, wow, okay, no pressure. But the RMI program has been super supportive in funding these eco-glider baseline surveys. So we've been going out we're in our second year of seasonal, actually greater than seasonal resolution deployments of gliders, and we're monitoring. I'll show some images of that in just a little bit. But it's really meant to do this baseline monitoring for the oceanography, but also the ecology, using all those cool biological sensors. And it's really meant to support the offshore wind planning process in the region, but also to help with filling in those gaps for the baseline early so that we can start thinking about climate change impacts. And then another one was led by Josh Kohut, who I had mentioned previously, was focused on using gliders with passive acoustics to look at presence, habitat use, seasonality, and migration patterns of the North Atlantic right whale. And that was a collaborative project funded by the Ørsted Ocean Wind Project. So those are just 3 examples of programs that we have been doing and some that are still ongoing in the Mid-Atlantic region. So now to show you some fun data that we're seeing and to kind of bring home the importance of understanding what our ocean is doing. So the first thing is the seasonal evolution of oceanographic properties. So we're really being able to start seeing the true variability of the system seasonally, but we're going out multiple years so we can start seeing that interannual variability. And there's a lot just from the last few years that we've been going out seasonally. We've seen a very different ocean from last year to this year, for instance. But even seasonally, we're starting to see these natural seasonal patterns. Sometimes we'll get early onset stratification in the spring, sometimes it's later. Does that have an impact on biology? Summer, we see that strong stratification where we start seeing that lower dissolved oxygen, lower pH, lower aragonite saturation states in our bottom waters. So that's kind of our season of concern in terms of things like ocean acidification. And then we see a lot of storms in the fall, so how does that mix up the water column? How is that attuned to biology? A lot of times that's where, if that fall overturn will trigger migrations for fishes and things like that. And then we have winter, very well mixed, very cold. But again, we're seeing that true characterization of the system, but also seeing the physics, the chemistry, and the biology at the same time. And one event I wanted to just point out that's also related to management is this observation of multi-stressor events, where last summer we had our bottom water, we had a lot of low dissolved oxygen in our bottom water, we had low pH, low aragonite saturation states in our bottom water. So we had kind of hypoxia a little bit and also ocean acidification. And there were reported fish kills during that time. We don't know for sure if it was associated with this event, but these are the kind of data that will help us kind of figure that out. And also allow us some predictive power of how anomalous was this year, because we have now data going back. New Jersey Department of Environmental Protection has been funded of glider deployments focused on dissolved oxygen for the last decade. So we can go and look at those data, develop climatology, and figure out when and where are we having these anomalous events. And then we can convey this information for management. This is just one example of how we do that for ocean acidification. And we develop these maps every year that allow us to look at where in the water column and over the Northeast Shelf are we seeing potential ocean acidification. And then on the right, it shows areas where we see levels that might impact certain species based on their sensitivity from lab experiments. So we can start mapping, again, areas where specific species might be responding to that or might be negatively impacted. And then in terms of the higher trophic levels, we have the active acoustics and the passive acoustics working together on these glider deployments. So we're able to start thinking about prey fields for things like marine mammals and fishes and figure out, are they co-located? So really one of our big questions is what are the physics driving the prey fields? And then are there prey field hotspots that might be impacting migration behavior, foraging behavior of these high predators, so the things like marine mammals. So we can start looking at that in both time and space using these data. And just to kind of go beyond the mid-Atlantic, gliders are ever present in our global ocean observing efforts now. So we have them everywhere, and if we can start outfitting these gliders with more and more sensors for biology, then we can really expand that spatial footprint. So thank you. Hope I wasn't too over time. I don't think anybody's keeping time. I don't see a timer. Oh, you are? Was I over time? Perfect. Okay, thanks.
All right, thank you, Grace. Our next speaker, Christine Knauss, are you online?
Yes.
All right. So Christine is a policy fellow at Maryland Sea Grant. She got her BS at UMaryland in chemistry and biology and then worked at Horn Point, where she did her PhD. And she is going to be talking about microplastics, which is what she studied in PhD, and how they affect oyster larvae, especially in Chesapeake. All right, go for it.
Yes, thank you. All right, can you see the presentation slide?
Yes.
So, thank you for the introduction. I'm really happy to be sharing this work, and I'm very sad to not be there in person with you. So, as Mike said in his introduction, I am now an assistant research scientist at the University of Maryland Center for Environmental Science, and this last year I have spent as a Maryland Sea Grant Science Policy Fellow. And so I was able to start working on this project that I've been wanting to do for a long time. My background is in microplastic research, and I've been wanting to add sort of a trash or 3D or plastic pollution indicator to the Chesapeake Bay and watershed report card for a long time. The QR code in the bottom right is a form to contact us. I'll just start out now. This is going to be a huge, massive effort. I realize that we need a lot of help, and a lot of you out there probably have a lot of information that could be, and thoughts and comments that could be really useful. So I have that there now, but I'll also have it at the end. So to start, maybe all of you know about the Chesapeake Bay and watershed report card that the integrated score regions pretty much based on the tributaries and different sections of the bay. And then looks at these different indicators that have been identified as important for that goal. So for this, is eutrophication and some other things, and then uses long-term monitoring data to create to have a number, create a threshold, what is healthy, how much phosphorus is enough or too much, and then compare that to how much is there. And then so you do that over time, and then you end up with a story. And what's great about the report card is it's very public-facing, policy, legislator-facing, where people can digest it and you can keep track over time. So the 5 major steps for creating a report card, and Ian, the Integration and Application Network, has done this now for a lot of regions. So what is the big picture? What's most important to this region or this river or this basin? What do we measure to go towards that? Key thing that we're really wanting to look at over time. What is the threshold? What is healthy? For an example, for aquatic grasses, what are we trying to get to? Is 50, kilometers squared a healthy amount? Maybe we decide on that, through experts and different things, and then how close are we to that? And then you look at the data overall for all the different regions, where you can find out what regions are doing better and dig into the data more about why, and then work through that. So starting with Chesapeake Bay, moved up, realizing that it was more than just the bay and just the water, that the watershed and the streams and everything in that, and the people too, as are very connected to the land. All of the things that we do impact the land. So the last few years we've included different indicators, social and economic indicators, to include that here. But you notice that there isn't any trash, plastic pollution, debris, or even other toxic chemicals such as PFAS, PCBs included in this report card as of now. One important thing to mention is that the report card really, the main basis of a robust report card and indicator is consistent data, monitored data over time. So going back to talking about debris and plastic pollution in the region, just kind of lay the land of our current understanding of the Chesapeake watershed. So this is really important for all of the watershed states. There's a ton of work that is being done through research programs, through all levels of government, through NGOs. I mean, people are very interested in trying to tackle this problem. There are dozens of microplastic studies and occurrence data in different environments. There is some trash monitoring and reporting already, such as in the Anacostia and the Baltimore Harbor. Most of the data that we do have is more aquatic, and meaning, aquatic, so water sampling, sediment sampling, and then aquatic organisms, so shellfish or fish, for microplastics in particular. Or shoreline. By shoreline, I mean, immediately next to a body of water. River banks, stream banks, and also, shoreline. We do have in the region some abandoned, lost, derelict vessel and fishing gear monitoring and removal. We do have some itemized bans in different regions, so microbeads, plastic bag bans, styrofoam bans, and some other policies. But air, soil, biosolid, drinking water sampling is definitely in its infancy in this region. So we have a ton of great work, but it's all because of the grassroots nature of how this, all this research and interest came to be. It's disconnected and why now? We have, some data, but we don't have, I'd say it's pretty clear that we don't have enough data to create a robust, indicator. We need long-term monitoring data. But I think right now is the perfect time for this. There is a lot of momentum. Like I said, there's interest in all the states by a lot of people. We are coalescing around methods and standards for many of the categories of the debris. And some are already standardized, like for litter monitoring. And to move forward more effectively, so we've done a lot of the groundwork and sort of individual work and small group work. We need to move forward more effectively. We need now a coordinated approach. And going back to the 5 sort of main steps of building an indicator, I think building a report card, I think we can also use this for building this complex indicator. And I think the last, a few years ago, we knew what the big picture was, but we weren't as sure how to measure things and what is healthy. And so I think now we have a lot more information for the second and third step is what and how to measure and what is healthy, and we're getting there. So our big vision for this, and we're just in the beginning, so please, if you have thoughts or ideas or anything, please reach out to us. So our big vision is to be, I'll start at the bottom, going from very small debris. We call it micro debris because it's not just plastic that we're after. You can consider tire wear particles, road wear particles, other man-made. That's where we chose that word, man-made particles, all the way up to derelict vessels. They're all important, and we think building this whole framework, building this big, ambitious vision is going to be the best in the long run. And so these are just some examples of things and their sizes. Now the top part of this diagram is all of the different places where we think that we can be sampling and monitoring and looking for these different debris. So I'll start on the top left, wetlands and beaches and those shorelines. We're already doing a lot of cleanups. There's data coming out from there. Looking at the benthic species and seagrasses, we like to look at the sediments. We think we can monitor there. In the water column for all different types of sizes, and then also in organisms. We'd also like to look at airborne microplastics. So I've talked to a few people at NIST who are beginning to look at that, PM10, PM2.5, monitoring the area. And then the last one that's not on here is, so I'll highlight this, is wastewater treatment. So wastewater treatment and biosolid application. I think wastewater treatment is a great place where we can be testing drinking water, the biosolids, and the effluent that comes out. So that's sort of our vision, is to take all of these together, all the data that would come out of monitoring programs, put them together, be able to come up with numbers, and then eventually have a score. So how I see this happening, so this is kind of our first steps, is establishing the different categories, by sizes, by item types, look at the different units, how we're going to do that. Gather and evaluate the existing data. We're going to need a certain level of quality of data, and so evaluating that. Identifying all of the monitoring gaps. There's definitely, we're not monitoring everything that I just showed on the slide before. So what aren't we monitoring? And then there's some beginning tools like the Toxicity of Microplastics Explorer. We can use the total maximum daily loads for trash to start evaluating what is the healthy level or an acceptable level of all of these things. And then so that's sort of where we are now. And then further down the line is to create a framework to fill those monitoring gaps. And there's been some good work from Tetra Tech, Bob Murphy at Tetra Tech, the Marine Debris Program action plans for the Mid-Atlantic and Virginias and a lot of the states in we're doing this and this takes time, there's going to be better risk evaluation and the effect threshold, so we can come back to that. And then eventually, we will be able to create a score, but it takes a lot of work, a lot of groundwork to get there. So a few examples of monitoring programs, and this is not a comprehensive list at all, just a few. So the Baltimore Harbor trash wheels, there's 4 of those. There are trash traps in the Anacostia. The Anacostia and the Baltimore Harbor also have trash total maximum daily loads. Prince George's County, Maryland, is the stream trash monitoring. Maryland and Virginia, there may be other states as well, monitor some of the abandoned lost and derelict vessels. And there are many, many nonprofits across the region that are doing, shoreline cleanups, other cleanups, and they're registering this data in different apps. And so those are all places where we can get data from. So I know there's a huge amount of challenges for this. It's a huge suite of categories, and so it's going to take a lot of coordinating across the region and across many different groups. And many of those, like I mentioned, are not monitored yet in a small region or across the whole watershed at all. The methods still need to be developed for monitoring and automating microplastics analysis. Another part of my job is working on a project that does that. And then really important is the QA/QC of all the data, and there are existing guidelines in other places that we could use, but we should, we will be making guidelines of the quality, what is the level of quality of data that can be included in this. And then also, the risk levels in some ways are starting points, but they're not well defined. How many boats in a river is too many? Or, how many, we don't know yet how many microplastics in sediment is too many. So the last thing and the most important thing is please help us. This is, like I said, a huge undertaking. The QR code, is for a contact form. You can email me. And if you'd like to learn more about the report card or the newsletter that we published with that about some microplastics in this indicator, you can find that at Ian at UMCES.edu and their publications. And then just a big thank you to Lily, Nicole, and Vanessa. We have all been working on this for the last 7 months, and it's been a lot of work. So thank you to them, and thank you everybody for listening.
Thank you, Christine. All right. The last presenter of this session is Brooke Van de Sande. Had to travel quite a long way to get here. She grew up in Toms River and is now a whale naturalist at Jersey Shore's whale watching. But more importantly, she is a student here at Monmouth studying marine and environmental biology and policy. And as a little addition, she was recently this year at the Southeast and Mid-Atlantic Marine Mammal Symposium at Duke. And she was awarded the Best Undergraduate Student Speed Talk presentation. So congratulations. So without further ado, Brooke, you're up.
Thank you for the introduction. I know I'm the last presentation before lunch, so we're going to keep this short and sweet as possible. So thank you again. My name is Brooke. I am a student at Monmouth University, and I'm also a naturalist on Jersey Shore Whale Watch out of Belmar, New Jersey. So we're going to jump right into it. So I've been researching the humpback whales along New Jersey for the past 2 years now. And my research is about identifying the relationships between visual sightings of humpback whales and then using environmental DNA to see if there's any relationships. Ooh. It's not going to the next one, sorry. Okay, perfect, thank you, sorry. Okay, perfect. So just to kind of give a little bit of background information, there has been an unusual mortality event along the Atlantic coast for humpback whales that was beginning in 2016, and it was declared by NOAA in 2017. Now, there were many necropsies that were conducted on about half of the stranded whales, just because of access issues. But for 40% of the whales that did have a necropsy conducted on them, about 40% had signs of human interaction, such as vessel strikes or entanglement. So this is a really new and an ongoing problem. So just to kind of give like a mapped visualization of this, so this is the New Jersey, New York UME from 2016 to 2024, and these are just all of the humpback whale strandings that have occurred kind of along the New York Bight area since starting in 2016. So we can see that this has really been an ongoing problem, and there's kind of more annual whale strandings that need to be solved and really figure out what the problems are. So as we all know, the effective conservation and management of marine mammals is really derived from the continual monitoring of their habitat use and their migratory patterns. So researchers and scientists use a few main monitoring techniques. Of course, visual surveys from boats are the most common. We have passive acoustic monitoring as well that's really common, or PAM monitoring. We have aerial surveys as well, but these are kind of being a little bit replaced by like UAS systems or unmanned aircraft systems like drones to conduct aerial surveys. And then we also have satellite tagging as well. But of course, all of these monitoring methods have their pros and cons. And more recently, a new non-extractive way to really detect and monitor marine species along the coastline is eDNA, or environmental DNA. So eDNA is simply just the genetic material that is shed or released into the environment through an organism's hair, its feces, or its skin cells. And it's very non-invasive, and it can be the water samples can be easily extracted right from the marine environment. And eDNA's main abilities are really just species detection and then being able to do that over very large spatial and temporal scales. So kind of just a little background, so eDNA for marine mammals. So eDNA can really be used as a complementary monitoring tool with other techniques, which just allows for more accurate data. Instead of using one monitoring technique, we can combine multiple, and eDNA makes it really simple to do so. And a couple other research studies, specifically this one, Alter et al. 2022, actually had successful detections of marine mammal DNA with visual sightings, so my research kind of just takes that to the next step. So to give you guys a little background information on the local New Jersey and New York humpback whales, so over the last decade, there has been an increase in nearshore visual sightings of juvenile humpback whales. Now, this is a very recent phenomena, which means there is a lack of extensive research for why the whales are showing up along the New York Bight and really how they utilize our coastline. And of course, you guys all know this area is a very highly urbanized region. So the whales that are staying here for extended periods of time are highly vulnerable to anthropogenic interactions, such as vessel strikes or vessel collisions. And most sightings of the juvenile humpback whales, specifically inshore, have included foraging behavior on Atlantic menhaden, which is their primary food source around here. And we have seen increased menhaden populations over the past decade, which has coincided with increased nearshore visual sightings of juveniles. So this could mean that this area is becoming a possible supplementary feeding ground for these juveniles. So my main research objectives were to confirm whether eDNA can just detect and identify humpback whale DNA along the New Jersey coastline. So that was my first objective. My second one was to compare eDNA to confirmed visual sightings of whales and then determine if there's any possible relationships. And my main hypothesis was that humpback whale eDNA reads would be higher in water samples that I took when there was a confirmed visual sighting of a whale compared to the samples I took when there were no whales in the area after continual monitoring. So before I was bumped up to a naturalist on Jersey Shore Whale Watch, I was an intern last summer. And off of Belmar, we conducted non-systematic boat sampling, like I said, from Jersey Shore Whale Watch tours. So when I was an intern for last summer, what I did was I conducted 2 samples on every trip we took, when weather permitted. So I took a water sample just using a simple bucket. You can kind of see me up there just kind of hanging off the side of the boat a little bit. So I collected a water sample when there was a confirmed sighting of a humpback whale less than 100 feet away. Of course, when weather permitted, or of course we were prioritizing the safety of the animal, we tried to get as close as we could, but we tried to keep around 100-foot distance from all of our whales. So I took a water sample with a confirmed sighting, and then I took a water sample when there's no confirmed sighting, so this kind of represented my control sample. I filtered my water samples right on the boat using a vacuum pump right then and there, so it made my process a little speedy and a little quick. And then I brought all my samples back to the lab where I conducted DNA extractions, PCR, and then they were recently sent out to the University of Maryland for metabarcoding sequencing. So this is just my water sample locations. So I sampled from June through August of 2023, just when I was interning on the boat. And the blue dots represent samples I took with a confirmed sighting, and the pink dots represent samples of no sighting. So my study area was just limited to where we go on Jersey Shore Whale Watch's tours. So we typically start from Belmar, that's where our boat is docked, and then we go all the way towards Sandy Hook, and that's kind of our range that we typically follow. So I recently got my results back, and I was able to do a really preliminary analysis of what I have. So what I found was I used bioinformatics in R using a package called DADA2 that analyzes all of my water samples, and I found that 5 eDNA samples all came back positive for humpback whales, and of those 5 samples, all of them corresponded with visual sightings. So that's a really good positive relationship that we were looking for. And then also kind of just shifting a little bit, but something I thought was really interesting, 14 of my eDNA samples also came back for positive for bottlenose dolphins, and 6 of those were visual sightings, and 8 of them were corresponding with no visual sightings. So I thought that was really interesting just to include because it really shows eDNA's capabilities to detect marine mammals even if they're not even visually sighted there. So it's a very strong and sensitive monitoring technique. Before our future analysis, I'm going to be kind of deep diving into that over the next few months. I want to really focus on relative read abundance, which is really just the percent of the species-specific whale DNA within every sample compared to the total DNA. So like I said, I'm looking that there would be higher eDNA read counts on samples I took with a confirmed sighting because there was a whale in the area. And then I also want to determine if any other environmental factors might affect the environmental DNA in my samples, such as whale's location, its behavior, or water temperature and weather as well, because I kind of recorded all of those different environmental variables as well. But kind of just to wrap all of this up, eDNA is a really new and emerging field that really just has so much potential, and my research really just skims the surface of its capabilities for effective conservation and marine mammal monitoring. But my results from this research will really help inform better management decisions for conservation efforts, so we can ultimately ensure that these humpback whales that are showing up along our coast have future protection. And then just thank you to everybody at Jersey Shore Whale Watch, all of my professors here at Monmouth, and the MEBP program. Thank you, guys.
All right, I'm going to actually start off the question session with actually a comment. I'm just curious if you've used the data sets. It goes back to my MARACOOS work, that the Oceans Map portal for the MARACOOS data has sea surface temperature data going back several years, ocean color data, plankton, phytoplankton bloom data going back years. It also has HF radar throughout the mid-Atlantic. And those are measurements of surface currents every hour, every 6 kilometers or so from Cape Cod, Cape Hatteras. So you can look at where things are moving to. So you can actually get an idea of where things were going back in time, because these are measurements, right? So just curious if you've used any of that and any of your research yet.
So I haven't yet. I haven't really gotten to I just recently, over the past few months, did our preliminary analysis. I'm doing this work for my honors thesis, so over the next few months, I'm kind of finalizing everything. So I haven't looked at any environmental parameters yet. When I was on the boat, I collected my own sea surface temperatures. I also used the Beaufort scale as well for doing the different sea surface and the weather of the ocean and everything like that. I kind of did just basic environmental variable testing while I was on the boat. Just because taking the water samples and filtering it kind of took a lot of my time. But I am looking into using that for extra, to look at different environmental variables that I couldn't necessarily collect while I was on the boat, restricting my abilities as an intern. So yeah, I definitely would look into that for using that for my environmental analysis.
Be happy to help you out. Everything's out there. Everything
Yeah,
we
perfect.
do within MARACOOS, the data's put out there. So, but be happy to help you out. All right, questions from the audience? Cindy?
Can't help myself. Dr. Saba, I'm very interested in what you feel would be a good baseline time frame. The ocean is so dynamic, as you said, and species come and go, and there's just so much out there. How do we really? What would you characterize as a good, strong baseline data set over in terms of years and how many seasons, things like that?
It's a really, really good question, and we've been asking that ourselves. There's just, yeah, you're right. There's just so much variability. When you think in terms of interannual variability and detecting what that is, you'd want at least 5 years of data, right? 10 would be better. When you think of long-term trends, so ocean warming, ocean acidification, on that scale, those consistent rates of change, you're going to want decades. So 30 plus years, 50 even better. it's hard to come by, right? But when you're thinking of like localized, we're doing very localized sampling off the coast of New Jersey, and we've been deploying gliders. Rutgers has been deploying gliders for decades now, almost two.
Just about
More
two,
than
yeah.
two, almost two. I don't even know. It's
Exactly
been a long
two.
time.
Exactly two.
Since I've been there anyway, from 2010. But, and so we have all these data, we just may not have it at the resolution that we have it now. So our resolution is getting better. And so I can't specifically answer your question because I think we're still asking that ourselves, but we're at the point where we're gathering at least what we think is sufficient data to understand seasonal patterns, typical seasonal patterns, so developing things like seasonal climatologies. And we're hoping to be getting at that interannual variability soon with the density of data we have recently. So we're getting there, but it's a great question.
Mike, we have a question from online, and then we'll go back into the room. But I want to ask you this question first. So how many times when there was a visual observation of a whale was there not eDNA?
So, So what I did was I was able to do a really brief preliminary analysis. So there's other packages in R and other statistical analysis that I could do that could extract more DNA from those samples. So what I just have now is that 5 of my, I think 35 samples came back positive for humpback whale DNA, and those did correspond with visual sightings. So I don't have any. None of my samples that came back positive for humpback whale DNA weren't with any visual sightings. So that's kind of, I can dive a little bit deeper and do a little bit more extraction, and that's kind of what I'm working on now to see if I can get a little bit more results out of this.
Yeah, I think the question is if you had a visual sighting, did you have any situation where it did not come back positive for eDNA?
Oh yeah, so far what I have now is I think out of my 35 samples, 15 or 16 were taken with a visual sighting, so only 5 came back positive, so there would probably be about 10 or 9 of them that there was a visual sighting and no DNA found so far.
Thank you.
No problem.
And then we have one in the room, and then we'll go back online.
This is also for you, Brooke. So just in the realm of eDNA in general, I'm relatively new to it, but I imagine you've been in the literature learning about it, obviously working with it. When using this as a parameter to understand who may be present, can you just elaborate some on the restrictions of the data, what it can tell you, what it can't tell you, and maybe other parameters that are important to pair with it? Visual sightings being one, but when that's not available, just any elaboration you can offer there is helpful.
So I think eDNA, like I said, it's really newly emerging, so there's a lot of like any monitoring technique, there's the pros and cons to it. What I found really interesting, and kind of that I'm noticing now in my data, to kind of keep this short and sweet, the specific filters you use when filtering the water for DNA really influences how much DNA you get actually out of each water sample. So I use a specific filter, and then it was just the one that was recommended in the literature. And then we are finding out, we're actually conducting studies in the lab right now, that those filters aren't even the best ones to use. So we're kind of like, oh no, we should have used these other ones, because those actually collect and extract more DNA from the specific water samples. So there's limitations pretty much for eDNA based on the kind of equipment you use and the process you follow for extracting the DNA and filtering the water properly. So that's kind of just what I'm learning now going through this process and looking back, if I did more water samples maybe next summer during our season, this is what I would do differently. But eDNA is really interesting because like I said from my results with the bottlenose dolphins, we had about 15 samples that came back positive for bottlenose dolphin DNA, and about half are when we didn't even see them. So it really just shows eDNA's capability to detect marine mammals, even if they're not even visually observed. So that's why it's a very non-extractive, non-invasive way. You don't need to actually go out, tag them, or deploy monitors to actually listen to them, or just visually sit on them for a while and watch them. You can just go out and simply collect water samples, and I was just trying to see if you collect a random water sample, if the higher the DNA in that water sample is, could that represent that there was a whale in the area even if you didn't see it? So that's kind of what I was looking for, because it's a very non-invasive way to monitor them and really track their movements along the coastline without even really needing to see them. So there's, it's really good and it could be really beneficial, but then again, it kind of just depends on the kind of process you use and the equipment and the exact method you use. I'm kind of just figuring that out now. No problem.
I think there's one more question online. I know we're heading toward the end of this. Mike, are you okay if we do one more? Okay. Okay. Do you know what tissues or bodily excretions from whales provide DNA samples? Example, mucus, respiratory fluids, etc.
Yeah.
Oop.
So I don't really know the exact, it's hard to like pinpoint exactly what like specific DNA we're looking at, but I just know that the primary things that they look for, like when using eDNA, is like different like hair follicles, skin cells is really important as well. And then people are actually doing something really cool where they actually are using drones to capture the air when a whale surfaces and breathes and it exhales at the surface. They're actually collecting just their breath and like the DNA just from them exhaling at the surface, collecting that DNA and then using that to filter for whale DNA as well. So that's something really cool too. But I don't know exactly like the specific kinds of, bodily fluids or mucus or anything. I just know that it's pretty much, in the water. Even if we're swimming in the water, our DNA is going to kind of shed off of our body as well. So it's pretty much just the same kind of simple thing.
One more. So the question was, how long is the DNA maintained in the ocean?
Yeah. So the specific study that I cited earlier, Alter et al., when they compared visual sightings with the DNA found in the water, they also did like a longevity study as well. So they found that about 30 to 45 minutes is when the DNA starts to degrade in the water. So that's why it's really important, because if you take a sample and you bring it back to the lab and extract it, and you see that there's high DNA read counts for humpback whales, you could say, oh, there was a whale in this area, or the DNA from a specific whale from as short or long ago as 30 to 45 minutes ago. So it's a really interesting study to find, to see oh, there was a whale here recently, even though we didn't just visually see it.
Great.
All right. Thank you, everyone. Thank you for presenting.
Just two thoughts. Thanks to the panel. I did on the marine environmental DNA. I can help myself a little bit, but just to say, her professor Jason Edoff has really been one of the leaders in this, and almost every question that was asked is being actively engaged around his colleagues in marine environmental A. I'd like to shout out Urban Coast Institute. Actually, about 6 years ago, hosted with Rockefeller University program for university the first marine environmental, and there's a lot of data, Will, available about these questions. All the questions you asked have been questions the eDNA research community have been asking themselves for the last 6 years pretty intensely. There's a very strong effort right now with Smithsonian and others. A national marine environmental DNA strategy was just released about 6 years ago, 6 months ago. So there is actually a very proactive commitment to developing these issues and asking the questions. There's already one project that Jason did with Rockefeller around working with fish trawls and trying to figure out whether this is substitutes for that. He worked with a Rutgers graduate student, I think, to actually match some of the eDNA data with some of the physical oceanography data to be more predictive with regard to what the sources of that eDNA. So anyway, lots of cool stuff about eDNA. I want to say lunch we're going to go to. I want to say, everything we're talking about right now, looking to the World Cafes at the end, MARCO has a marine debris work group. MARCO has an issue with regard to ocean conservation. It has an offshore eDNA MACAN thing. So all of the issues you're hearing about in the specific efforts really do flow into a lot of the commitments that the MARCO work plan has already engaged on and the Mid-Atlantic Ocean Policy Committee is looking at. So do think about what you can bring to that discussion tomorrow, because is there a way for us to take that back and actually incorporate into our work moving forward. So lunch is a little abbreviated, but you have an hour or so. So around 1:45, we'll be gathering you all to come back in for the next session. Thank you.